Okay, this is a screencast for the butterfly group. Since I did a screencast covering the statistical analysis for the water bug group, I felt like it was only fair that I do one for the butterfly group as well. So you've already been exposed to this, so it shouldn't be too complicated for you, hopefully. But basically, in somewhere on your computer, you should have a butterfly analysis script that I gave you. Um, and you should have the butterfly file um, in the same directory somewhere on your computer, hopefully. So let this load up, and so here we are in the butterfly analysis. First thing you want to do is you want to get the WD, get your working directory. And my working directory is in lab materials down here within the biology 2019 Biology 133 folder on my computer. That's where it's held. There are a bunch of things that you will probably need. You may not have some of these, so as you're loading these packages, check your error messages down here in the lower left to make sure that they're actually loading. If they are not loading, you can go over to the panel that is on the right-hand side and go to Packages up here and click on Install and then type in, for example, you might not have em-means. Type that in and it will go and load it when you click on Install. And then once it's installed, you can then load the library. So once you've done that, click on this little arrow there, and you can read in your butterfly data file, and you can make sure that it read it in correctly. Now you see down here that all of the air temperatures are in degrees Fahrenheit, so we need to do some cleaning up of the data and some conversions. So first thing you want to do is you want to check and see if these data are in the correct classes, if it's the right kind of data. So because you've numbered your individuals should be integer data. Species should be factored data because it's names of species. You can check the levels to make sure that there isn't any weird misspelling or anything. You should only have 3 levels: Coleus fluidice, Danaeus plexibus, Vanessa cardui. You can check, once again, orientation. It's a factor. Its levels should only be horizontal and vertical. Temp type should be a factor, and it should either be air, butterfly, or net. No other ones. And then air temp in Fahrenheit and temp in Fahrenheit should both be numeric. So that's what the data look like. We can do the conversions. We're just going to make two new columns, one is CA and one is CB, to convert air temp from Fahrenheit to centigrade. And when we do that, it should now have 2 new columns that have considerably lower numbers because you've taken 60 degrees and you've converted it to 15.611. On the centigrade scale. So let's go ahead and set ourselves up a formatting block, and then let's see if the temperature types differ. So you went out and you took 3 different temperature types. You took air temperature, you took body temperature, and you took net temperature. And if butterflies are regulating their body temperatures at higher temperatures than air temperature, then you should see that there's a difference there. We'll do this with an analysis of variance. We're going to use the linear models package where we look at the actual temperature, whether it varies by species, whether it varies by orientation, and whether it varies by temperature type. And what we should see when we do this is that the only thing that changes is temperature type. Temperature type is the only thing that shows any kind of variation. So we can get the summary statistics for this, where we look at the measured variable, which is temperature, and we look at it across species, across orientation, and across temp type. And when we get this, we see that the temperature type should be the only thing that really matters, and we see that air, butterfly, and net, butterfly is slightly higher, air, butterfly, net, butterfly is slightly higher, air, butterfly, net. Butterfly is slightly higher. Air, butterfly, net. Air, butterfly, net. Butterfly is slightly higher. In all these cases, it looks like, well, not in all these cases, but in a lot of these cases, the butterfly is slightly higher or significantly higher. And so that kind of gives us some insight into why that analysis turned out that way. We can look at this by making a plot of these things where we're going to plot the data that is in this big matrix down here, and we want X to be species, we want the Y to be the temperature, we want the color to be temperature type. And when we do this, it should give us a figure that looks kind of like this. And it's clear that the type of temperature that you're looking at, butterfly versus net versus air, is having a difference. The net temperature is pretty low, the air temperature is higher, and butterfly temperature is higher still. And those things are relatively parallel across the different species. The ordering of those three things is roughly the same across three species. And so that's why that figure is coming out as being significant. So, but we know that when we collected coleus, the coleus were at overall higher temperatures, air temperatures, and because they were collected at higher air temperatures, the butterfly and the net temperatures were also higher. And so it would be nice to be able to look at these correlations. And so the first thing that we need to do is we need to subset our data so that we have a revised data set that only looks at the butterfly data. And so what we see is a thing that if we go down here, under temp type, the only temp type that is here is butterfly. And so it's got butterfly, but it's got air temp and regular temp. So C sub A is air temp, C sub B is body temp. And so once we have this, we can go back to our water bug analysis. That thing that I just did just took the data and showed it to me in this window. Go back up to the top and we can get back to the not the water bug analysis. Let's close the water bug analysis. We can get back to the butterfly analysis. So we have those data. Well, now we can look at those data where we look at the effect of orientation on butterfly temperature. And what we'll see when we do that is we should see something that looks like, and then we're going to facet it and get it to facet it out. And we're going to basically see how air temperature is correlated with body temperature, but for the three species and according to orientation. Is it a vertical orientation or is it a horizontal orientation? So what we see is that there's a correlation between body temperature and air temperature, but the Vanessa cardui seem to be at somewhat lower temperatures than Danaus plexus or Coleus phyllodyce. And so that once again tells us some things about what we should be expecting. But because there's correlation between body temperature and air temperature, we have to explore this relationship somewhat. And so we can do that with a linear model and see if that relationship is significant. And when we do that, what we basically find is that body temperature is significantly correlated. The slope value is 1. So basically there's a 1-to-1 relationship between body temperature and air temperature, which is important. And I've placed down here what the equation is, what the R-square value is, what the F statistic is, and p-value is 2 times 10 to the minus 16th. It's less than 2 times 10 to the minus 16th. So knowing that that's true for body temperature and air temperature, it will also be nice to know if that's true for net temperature, so we can subset the data once again so that it just gives us net, and we can do the same thing. And what we should find when we facet that is we have a similar thing going on. And in this case, the Vanessa cardui temperatures are not any cooler than the other temperatures, so that might actually mean that something is going on among species later on. So we can test this relationship, and once again, we find that this is significant. The slope is once again practically 1, and so air temperature is the same as net temperature for the most part. So now we can do a two-way ANOVA, knowing that this isn't going to be how we're going to finally analyze the data, but we can look at the effect of species and orientation on body temperature. And when we do that, it looks like there is an effect of species, and we can get the means and confidence intervals of all of that. And what it looks like is Coleus is generally warmer than everybody else, but Danaus plexippus varies somewhat depending on whether it's a horizontal Danaus or a vertical Danaus. And so we can plot this once again, and we get a plot that looks like this, where it looks like Coleus is the warmest. There's some interaction here with Danaus plexippus, having big differences between horizontal and vertical. And Vanessa cardoi, there's no difference between horizontal and vertical, but they're generally cooler. And so it's really the species effect that is coming through, as you can see from this thing. But because we know that we collected Coleus at warmer temperatures, as we can see from this plot here, then we have to remove the effect of air temperature that they were collected at. So we can go back to this butterfly data set that we had before, and we can do an analysis of variance. And what analysis of variance does, it looks at body temperature as it is correlated with air temperature. We remove the effect of that correlation between body temperature and air temperature, and then we adjust those values, and then we look at orientation and species as main effects. And so when we do that, we get something that looks like this. And once you remove the effect of air temperature, all of the other main effects, orientation, species, and the orientation by species interaction, are no longer significant. But do take note that your orientation by species interaction is almost significant. What does that mean? So I wrote up here, it's on the cusp of significance. And what that means is that we have to use least squared means to calculate what the adjusted means are, to calculate what the average body temperature would be after we remove the influence of the correlation between body temperature and air temperature. So that's kind of a complicated thing here, but we just go and get the adjusted means from the analysis of covariance analysis that we did earlier, and we want to get adjusted means divided up by species and orientation. This is not a data frame that it is produced down here, so we need to convert that into a data frame, and we need to then make sure that our levels are all there. So we have the three species twice, because we need the three species for both horizontal and vertical, and then we need to know that there are three horizontals and three verticals, because we have three species. And then we need to calculate the width of the confidence intervals from the lower limit and upper limit of the confidence intervals. And so we can just use the lower limit and subtract it from the mean. That will give us the width. And we're going to put that in a new column in this new data frame. And so what we're going to do is we're going to do that, and you can see that now there's a confidence interval over here in the far right-hand column. And then we can plot that, and when we plot that, we see that things are different because of this correlation with temperature. And the reason that your interaction is almost significant is due to the fact that Danaus plexippus is warmer on average than in the horizontal than in the vertical, and Vanessa is warmer in the vertical than it is in the horizontal. So painted ladies are warmer in the vertical than horizontal, and monarchs are generally warmer in the horizontal than in the vertical, and there's really no difference between the two orientations for Coleus. So that's telling us that different species may be thermoregulating in different positions, different orientations, but it's not quite statistically significant. So really and truly, if you're going to say this in the strictest sense, there's no difference among these things, and there's no significant interaction. So then we wanted to know if there's an effect of temperature on operative models. And the operative models are, in theory, a model that will gain temperature, but it doesn't have the ability to behaviorally thermoregulate. And so It's kind of like what the temperature would be for a static organism just laying out there in the sun. So you guys went and gathered operative model. You put it in a data file. The data file that I have it in is called models, and then I'm going to just leave that as models. Once again, it's all in Fahrenheit, and so I'm going to check these things to make sure that they're all the correct types of data, but then I'm going to convert the Fahrenheit data to centigrade data. And then what we're going to do is we're going to basically just conduct a simple analysis of variance to see if there are differences between the model types and air temperature, between the model types between air temperature, vertical temperature, and horizontal. There are differences, and we can do a Tukey's analysis of variance where we then test to see what's different from what. And horizontal is different from air, highly significant. Horizontal is different from vertical, but vertical is not different from air. And we can look at what that looks like, once again, by getting the means. Getting the means. And you can see that the horizontal is much warmer than the air or the vertical. And then when we plot that out, it's going to give us this figure, which shows that. If these butterflies were tethered down in the horizontal position or in the vertical position, they would be very different temperatures. But when we actually measure actively thermoregulating butterflies, these butterflies are virtually the same temperature. And so they appear to be thermoregulating in a way that keeps their bodies cooler than they would be if they just were just out there in the horizontal position. Unfortunately, these things are all probably correlated with one another. The horizontal ones are probably warmer when the air is warmer, and so we could actually look to see if that's the case. And when we do this, what we essentially see is that air temperatures are correlated with model temperatures, but the places where they are different between whether they are horizontal or vertical, and so we can actually just separate those things, and you can see that the correlations are there. It's just that the horizontal models are much warmer than the vertical models, but then within those positions, they are correlated somewhat with air temperature. And that's pretty much it. That's a quick run through of what your data mean. Now, you don't necessarily want to use all of these figures because some of them aren't really relevant. So, for example, you had an ANOVA, but the main purpose of me showing you the ANOVA data is so that you can see how the analysis of covariance then changes the result from the analysis of variance that you had before. Keep in mind also that these are done with circles, so if you print in black and white, then these two circles are going to look the same, so it's going to be difficult for me to tell what's horizontal and what's vertical. And so you might want to print your final version in color so that you can see those differences. Remember that you can export things as PDF models, as PDFs, and I would do most of yours, I think, in landscape. And so you can call these, ANCOVA results. And save that. And when you save that, it gives you a figure like this. And when you have a document that you're writing, like your lab report, you can basically get rid of these data, and you can essentially just go up here to picture. You can get a picture from file. You can go to whatever folder you have stored your stuff in, and once you've stored your stuff in that folder, I save that as an ANCOVA in the lab materials. So we have ANCOVA, and when we plunk it in there, that's what it looks like. And then you can write an informative caption that goes underneath here, figure 1, blah, blah, blah. Figure 1, give it a title, give it an explanation, and you can include the graphs right in the text of your results section. So hopefully that will be of use to you, and once again, let me know if you have any questions.